So I'm going to talk today about next generation lipid nanoparticles for RNA vaccine and therapeutics. And the future is already here. As you can see, 23 years ago, Mr. Bean here already understood what's the difference between RNA and DNA, and he needs a good template, somebody to copy from. And the lady, the mother of dragons, already showed us what's the difference between double-strand DNA to a single-strand RNA here 10 years ago. So the central dogma of biology, basically going from DNA, to RNA and proteins, have been around for many years. And it's now the next generation that we target the RNA, and we want to target it specifically to specific organs, and within the organ, specific cell types. So we are living in the RNA arena now, that we can do many things. We can silence genes, we can activate them or express them, we can edit them, and in the very near future, we will be able to replace them. And what can we do with this? So we can silence genes predominantly with SI, small interference RNA. We can activate them with a messenger RNA, and we can edit them and replace them by many types of genome editing tools. For example, the CRISPR-Cas9 one that reached the clinic, and we have already a very nice clinical example, which is Cascavy, that is clinically valid as a new drug therapy modality. So the challenges in RNA therapeutics is twofold. There is the RNA itself, and I'm sure you already heard a lot about RNA impurity, but it's also the delivery issue. So we need an appropriate delivery system. And the requirements are as follows. We need an efficient RNA encapsulation. We need evading clearance mechanism, avoiding toxicity and immune activation by the carrier with the RNA payload. The carrier must internalize, and the RNA must be released. And then, of course, we need specificity. So what we're going to talk today is about lipid nanoparticles that being a major player in this. And we already have 4 clinically approved drugs and vaccines. The first one on Patro, an aniline drug, was approved with lipid nanoparticles and siRNAs already in August 2018. And the siRNA is encoded here basically against the TTR amyloidosis. And then we have 3 vaccines, the 2 COVID vaccines by BioNTech, Pfizer, and by Moderna, and an RSV vaccine by Moderna. We have 4 lipids, sometimes more than 4, in a formulation with a favorite RNA, and it has a lot of advantages. It's protecting the RNA. The system is completely biocompatible, biodegradable, protecting the nucleic acid, increasing the stability in circulation and therapeutic index. And of course, as I mentioned, some of them are clinically approved. In my lab, we are interested in combining first RNA delivery system, including also protein engineering, with new potential drug targets. So we know how to do many things. We know how to silence genes with RNA. We know how to activate them. We know how to edit our genome with RNA, and hopefully in a very near future, how to replace genes. As a vision, we are living in the revolution. And here we're going to hear a lot of examples today about purity, about the importance of being able to control your analytics, which is very, very important. But it's not only the delivery, as I mentioned, it's also the payload. As an example for that, we are collaborating in being able to open, close genes, or to be able to control their translation, and of course, their stability. In this sense, the integrity of the payload, the integrity of the coating with the lipids is super important. Another example that we recently published is the ability to use circular RNA as a new modality inside LNPs. But still, the open questions are endosomal escape, which is a bottleneck for LNP-mediated therapeutics, and understanding the intracellular immune response of lipid nanoparticles. All in all, we're going to hear today lots of talks about the importance of analytics, how we can control each step, and hopefully that will give us the next generation LNPs. I want to thank you for listening, and I have enjoying your time.
Hi, everyone. So, my name is Tristan, and I work in Sartorius BIA Separations, and I would like to sincerely thank everyone for bringing this together, and especially now to Dan for showing us and talking to us about really what are LMPs, what's their current status, and what's their future. And really, there is many things you can do with LMPs. And but you at some point you want to bring them towards commercialization, and then where you've done all the great work in biology and all of that, and then you want to really be sure that what you are getting is robust and reliable. So that's where you have to think about more intensely about the characterization and the other manufacturing challenges that you are facing with the LMPs. And when you do that, you face certain challenges, and people have faced them, and some vaccines and therapeutics that are already on the market, like Dan described, have at least partially solved those problems. But let's talk about what they are. So there is the inherent kind of instability of the particles, because LNPs are a supramolecular assembly that kind of all the weak forces, they make together the particle, they hold together the lipid envelope, and they hold together the interactions between the RNA and the lipids. But those are weak forces that are susceptible to their chemical and physical environment. And then that brings about the heterogeneity, which is a consequence of that, but is also a consequence of other factors on the molecular level. And then it's also a consequence of the stability and the storage of the samples. And then because these LNPs are very modular, so you can change out the ionizable lipid, you can change the lipid composition, and so on. That brings about diversity, that it's then, you can bring about subspecies of different LNPs, to say so. And you have to think about that when you're developing, when you're thinking about, let's say, the stability and the characterization of your LNP. And then because in a simple system of LNPs, let's say you have four lipid, so four different lipids, like Blaž described at the start, and then you have the payload, like one RNA. So you have 5 different molecules that are interacting with each other, and that brings about also their interactions, and on the molecular level, that can bring about degradation of each molecule, and then interactions of those further on. And all of that, even inherently, but also because of those stability issues, can give you complex morphology and not just very simple spherical particles that we maybe like to think about. Now, in characterization, then there's these challenges. Because of all those reasons, you have challenges that the analytical methods currently available are poor predictors of in vivo and in vitro behavior. And that generally means that what we are looking at is not really the most important part, or at least it's not the whole part. So that what we get as our analytical results, characterization results, is not the whole picture. Or we are changing the system too much by wanting to look at the particles. So that's why I'm saying here that for intact particles, there are very limited reasons on how to analyze them without any pretreatment or any labeling, because labeling is a very powerful tool on each level, on like when you have the particles formed before, or just for labeling RNA. So many different ways to label your species, especially with fluorescence, because it's very specific and it's very powerful. But we have to realize that all of that changes the system a little bit. Let's say if you're using fluorescent RNA, that might be physicochemically very, very different to what you might have if you didn't have that. And that's that's why we want to kind of strive towards non-labeling techniques like maybe the more developed pharmaceuticals are using. And that's why, because of all those reasons, we then have a kind of a complicated CMC, so chemical manufacturing controls for our drug product, and we want to strive towards solving that and having things that are applicable and that give us the results that can predict how our product is going to be acting. So the analytical platform that we have developed that has been described quite a lot in this video already, so I'm going to go through this quite quickly. But it's like a two-dimensional chromatography setup, and in the first part of this, the OH column is a hydrophobic interaction chromatography. So here we are utilizing the hydrophobic properties of particles such as LNPs. Basically, the larger the particles are, the more they stick to the column because they have a larger hydrophobic surface. And even then, if you compare them to non-spherical particles like RNA, that is also polyanionic, it doesn't bind to the column as well, so it gets separated from the LNPs based on their chemical properties. And then you detect both of those species with the appropriate detectors. So for the particles, for the LNPs, you would use multi-angle light scattering. So you just direct light scattering information by observing your LNPs. And for the RNA is the UV-Vis detector that detects the chromophores in your RNA. So the nucleobases are very UV active, and it's easy to detect RNA this way, but you do have to separate it from other things beforehand. And that's where the SDVB column comes in. So we separate it from particles in the first step, and then in the second step is reverse phase ion pair chromatography is the mode used in this step. And here is where you're utilizing the kind of you're ion pairing the phosphate backbone of RNA, and then you're utilizing the hydrophobic properties of that. Here also, the larger the RNA is, the more of those ion pairing reagent it's going to have on there, the stronger it's going to bind. So you're separating by size. But then additionally, not just that, you're also separating by hydrophobicity. And then you can separate away some other species that might be important in characterization. And just a quick setup of how this works. So you bind on the OH column the LNPs, you analyze them on the Maltz detector, and then RNA, you use free RNA, it goes past here and it's analyzed on the SDVB detector, while the encapsulated RNA then binds to the SDVB column. That's quite important, because this is kind of an online particle opening that we have developed. So when LNPs get eluted from the OH column, they bind to the SDVB column very strongly. And we figured out that you cannot elute them as intact particles because the interactions are too strong. So what you do is that you, with a high temperature of 60 degrees and an organic solvent, you basically dissolve the particles into its separate components. So this is how we can do with no sample pretreatment, because we are opening the particles on this column with the interactions with the solid state of the column and the percentage of organic solvent that we then use to do those separately. And then you quantify the encapsulated RNA by that disruption of the particles, and then you also can quantify the lipids if you put in another detector like the ELSD or the charged aerosol detector behind that. So the first parameter you can determine is encapsulation efficiency by comparing those. That has been described. And then a very important thing is that if you have co-encapsulated species, you are going to be able to separate them, which is quite impossible with like an assay-based determination of encapsulation efficiency. And this is not just important for encapsulation efficiency of those, but also for their integrity and for any other impurities that you might be forming. And for this, it's basically necessary to have LC-based analytics, chromatographic ones, so that you're able to separate those and quantify them. And then very important in applications for gene editing, like Dan described before me, for kind of CRISPR applications, where you need to separate away at least your Cas9 mRNA and your small guide RNA. And that is possible to do on a setup and with a chromatographic approach. Then in terms of multi-angle light scattering and the kind of particle size and its integrity and so on, we have different methods that can be applied. So here we have, for example, NTA, DLS, cryo-TEM. Some of them, like cryo-TEM, are very powerful, but they are very laborious and kind of hard to implement as a critical quality attribute, also not very quantifiable. Now, DLS is the most widespread one. It's quite good for, you get very fast results, but you can get very misleading results, because a very large particle will give you a much larger response, and that can kind of skew your results towards larger average values, because you are not separating subspecies. So that's why it's also good to do subspecies separation with certain techniques one might use. In this case, we can present the separation using hydrophobic interaction chromatography. So the larger particles bind more, and you get a sort of a separation. And you see here, if we see a plot of radius, we can see an increase by size as this peak gets further on. So we are separating these particles by size, and we can show that it's comparable to microscopic techniques like NTA. But importantly is that we also, by that, we remove away the larger particles, and then we can see subpopulations of particles. And here is an example of that. This could be obviously further, whatever you need, whatever you're expecting from your particles, you will want to change maybe those different ones, you change your method a little bit, so that you want to see some subpopulations that you're expecting or you think are problematic. Now, another very important thing, critical quality attribute of the drug product, is mRNA integrity, because if your RNA is not intact, you're not going to be able to get it into the correct protein that it needs to be translated into in your body. And with the larger your cargo gets, generally the more unstable it is. If we're talking about mRNA, and then, for example, saRNA, self-amplifying, is even larger, even more unstable. And then the smaller ones like siRNA are more stable in terms of that. But let's say if we focus on mRNA for now, it's quite unstable. So part of the reason, a major part of the reason why we are using drug delivery systems like LNPs, is that we want to protect it from that kind of instability. Maybe reduce the water around it, so reduce the hydrolysis a little bit, but then in the body also protect it from the RNases that will degrade it. And here, the important thing here we have on the LNP switcher is that we can separate away. This is one crucial thing that maybe if you're using capillary electrophoresis needs to be looked at. So if you're opening the particles and you have a lot of free RNA, or just even some free RNA, that one is going to be more unstable. So it will be more degraded than your encapsulated RNA. So you might get a value that's it's not real, and that will give you might then accumulate some different percentages of how intact your RNA is, and it doesn't correlate with your biological activity. Might be because of that also. So if you're separating the free from the encapsulated beforehand on some sort of a chromatographic system, let's say, and then you're using that to analyze the integrity, that would be much more precise and accurate in terms of doing that. So on a switcher, because we're separating the free and encapsulated RNA, this is inherent to the system. And then we have there's an article how to do this using reverse phase ion pair chromatography, to determine the integrity of your payload of each separate one. So separately for free and separately for the encapsulated. And you see for free RNA, this is like at high temperature, 35 degrees, you're increasing, it's very fragmenting very quickly, while for the encapsulated RNA, it's fragmented quite slowly. And you can see that it's more protected than the free RNA. Now, another crucial thing that's been going on in this field is the mRNA lipid adducts. Now, this was, I'm showing here from an article by Hashiba in this year, is that aldehyde impurities that are forming from your ionizable lipids, they can form as oxidative hydrolytic impurities during stability, or they can also form, they can also bring certain or similar impurities like that from your synthetic route of your ionizable lipid. So you get so you would get degradation, and then you would form aldehydes that can alkylate your RNA and can render it inactive, or can even maybe form certain side products and so on. So you can use, so it is quite important to use reverse phase chromatography to analyze species like this, because capillary electrophoresis is not able to quantify this, because they are, by size, they're quite similar to the RNA, but by hydrophobicity, they can be very different, because those long lipid tails attach covalently to your RNA. And here you see from the first article on this from Packer in 2021 at Moderna, you can see that the correlation between CE integrity and protein expression is not very good, while the correlation between reverse phase ion pair analytics and relative protein expression is very good, actually. So it's the integrity based on LC in this case correlates much better with the actual activity. And here we can see, this is an example from our analytics. We have a capillary electrophoresis on the top and SDVB, so reverse phase ion pair analytics on the bottom from the LNP Switcher. And we can see that by CE, the integrity of this RNA is 74%, whilst by SDVB is only 47%. But if we combine the intact part with this part, the adduct part, we get the same percentages here. So basically, those two are co-eluting in the CE, because they are similar by size but very different by hydrophobicity. So that's why it's important to monitor those by reverse phase ion pair chromatography. And this is very important for stability studies, because generally the trend would be to have some sort of a fridge stable LNPs. But if you are then, here you see we are increasing by temperature, we're increasing this percentage of adducts. So that is something when you're developing those conditions for stability, you will definitely want to look at, because at -80, those are much smaller than in a refrigerated temperature. So the LNP characterization is a growing field that demands new analytical methods that could be used to provide reliably and widespreadly. Some of the ones that are used right now are maybe limited in what they can give you, and maybe can give you some false positives or false negatives. So that's why you want to spread out and analyze your product further, but you want to use some certainly more advanced techniques, like I described before, cryo-TEM or maybe SEX or things like that, when you're developing, to really see how your product looks like. But then for further on, you will want to use methods that can be used on basically a daily basis for analysis when you're moving to a large scale commercialization of products. And that's why also here we're introducing and developing LC-based methods that have a long history of doing that, and are able to, especially are powerful for separating subspecies. And this is what this sort of a system can do for you. It can analyze a drug product, an RNA LNP drug product, from many different aspects. So I would like to thank everyone from our company for doing this work and for helping us with this work, especially our department, the PC4 Process Development Analytics Department, that some of them you have seen already in this session, and I really they are all responsible for this work. So really, all the thanks from my side to them. And then also thank you again to Dan Peer for who is a pioneer in this field, for presenting and showing us what holds the future for this field and how powerful these modalities can be. Thank you.